Okay. Hi, everyone. Uh, I'm sorry if there might be a couple of disruptions while I admit attendees from the waiting room. So for the next 15 minutes or so, I'm going to talk about a project that I've been working on in the past few years, and it involves understanding what exactly it is uh, for even two proteins to interact in a cell, and how this would then inform mechanistic explanations that are quite commonplace in cell biology. Protein-protein interactions really underpin a big part of explanations in cell biology, partly because this is an old adage, but um, because proteins are known as the workhorses of cells, and they do their function in part by interacting with other proteins. And understanding which proteins interact with each other really plays a crucial role in determining the mechanism involved in whatever phenomenon in the cell it is that we're trying to explain. Now here I'm showing a snapshot of a cell membrane, but proteins could be within the cell, could be on the membrane, they could be interacting across the membrane. But here I'm just showing, for example, a few proteins that are in proximity of each other, and they would be interacting with each other, with the lipids in the plasma membrane, with other small molecules. So lots of quote unquote interactions are taking place. But one thing is when you, and here I'm showing a more cartoonish schematic of um, a few proteins interacting next to each other on the cell membrane. When you dig deeper into what it is for two proteins to interact, in fact, um, 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 it's not quite clear in the literature what we really mean when two proteins are interacting. So obviously, we are borrowing a term from human language. Um, um, Interaction really um, comes about when we talk about, for example, two people interacting. But even in the human world, interaction isn't such a straightforward concept to understand. I mean, ju just the mere fact that two human beings might be standing next to each other at a bus stop doesn't mean that they're interacting. In fact, two people could be standing next to each other at a bus stop and they might not be interacting. And in fact, one of them might see a friend across the street, recognize them, and start a conversation with them, quote unquote, interact with them. So, this notion of interaction in the human world isn't such a straightforward concept. Now, this notion, or a rudimentary version of it, was transported to macromolecular um, research in the 1940s and 50s. And nowadays, when people look at protein-protein interaction, they just mean that in some kind of an assay or an experiment, two proteins might be next to each other for long enough for one of them to impart some physical, chemical, enzymatic change on the other, or vice versa. Both of them might be affecting each other. So there is a very consequentialist kind of notion when it comes to interaction in the protein world. But when you dig a bit deeper and ask, how long do two proteins have to be next to each other for an interaction to take place? What distance in the nanometer range Do they have to be next to each other for an interaction to happen? It's quite vacuous, in fact, when you dig a bit deeper in the literature. And many of the large-scale protein-protein interaction studies that are done, so here I'm just showing a schematic, but now we have more network-style types of interactome investigations. It all has to do with quick consequence-based readouts in some large-scale assays. And so it's really, I think, important from a theoretical biology perspective to define once and for all what a protein-protein interaction involves. Now, it could be a protein-protein interaction, a protein DNA interaction, protein small molecule interaction, any kind of part-part interaction. But just as a case in point, I'm focusing on protein-protein interactions right now. Let's quickly, so there are three columns here. Let's just take a look at the middle column. As I mentioned, interaction is a human concept. And in fact, I think a handshake analogy isn't too far off the mark. When two individuals want to shake hands, there is a process involved. So the two hands start to move towards each other. When the hands reach each other, the two hands basically acquire structures that are complementary. Then after this initial contact, the fingers, for example, close. Then there's usually a movement up and down, even though, there are variations, but this is just a very rough estimate. And then afterwards, there's a duration to a handshake. And then there is release and return to the previous status. Bear in mind, in step 2, one or both hands could have the shape of a handshake without having reached each other. So my hand could assume the shape of a handshake kind of structure without necessarily being in an actual handshake. So the structure of a hand is appropriate to, but not caused by the other hand or by the context. And this is very important. The difference between a causal situation and just an appropriateness situation. We can have a similar thing when it comes to proteins. So if you take a look at the column to the left, we could have proteins similar to two hands where the interaction surface assumes its particular structure when the two proteins are next to each other. Or you could have a situation where the proteins have already assumed a structure before they reach each other. And for each one of these steps, if you look at the column to the right, we can define an aspect to an interaction. So in a very rough way, the process of an interaction could involve 4 stages. We first have preformed or induced interacting surfaces. Then we have contact, and contact in quotation marks, because there's still a minor distance between residues, even when there is contact and quote unquote binding. Then there is some kind of a consequence, some kind of a physical, chemical, enzymatic consequence. And to all of this, there is a duration and an end to the interaction. So there's a lot more to say, but okay, even if we have these aspects, how is that going to help? It's going to help in this way. Let's go back to a non-biological example. When let's say we want to explain how a pendulum clock works, what do? We describe the mechanism of the pendulum clock. This is what the escape wheel does, this is what the anchor does, and so on and so forth. We could call that a mechanistic explanation, but maybe we can call it a mechanistic description. For us to have genuine explanation, we also, in addition to the how part, we also need to be able to answer a why question. For example, why does this part of a mechanism work this way and not some other way? So, in a sense, we have to answer these contrastive questions. Why this way and not some other way? Um, the way we do that, for example, in physics and with a pendulum clock, is to rely on laws. So, for example, we rely on the law of gravity or principles of motion or friction to say, for example, the pendulum moves in a curve-like manner and not in a zigzag zigzag kind of manner, because of, for example, the way the law of gravity is formalized. Obviously, that might not be satisfactory to some people. We might need to even go down to the quantum level. But these laws or principle provide some answer to the why question. And mechanisms and laws together can give us a kind of a more complete or a genuine kind of explanation. In other domains, for example, in organic chemistry, we also have an appeal to mechanisms and laws. So, for example, an organic chemist describes the mechanism or the movement of electrons across different bonds, and that's a reaction mechanism. But they also appeal to laws of thermodynamics to, for example, answer questions like, why does the reaction go towards the right and not towards the left? Or why is it endothermic and not exothermic? So we have mechanisms and we have laws. In cell biology, we don't have many cell biology specific laws. People weren't really looking for them. I mean, biology obviously has principles or laws when it comes to the population level. So we have principles of evolution. Or when it comes down to a molecular level, we have chemical or physical laws like thermodynamics or laws of friction. But laws or principles specifically posited at the individual cell level are few and far between. There's some scaling laws, there's some other kinds of laws, but they're very few because people don't have a specific program looking for laws or principles in cell biology. Each one of the four aspects of a protein-protein interaction could be explainable either using already existing laws from physics and chemistry or laws that are specifically posited for proteins in the cell. Laws that, for example, could describe the duration of an interaction between two proteins, or laws posited that explain the structure of disordered domains in proteins. So other than the fact that understanding protein-protein interactions is has value in and of its own, another benefit that it could have is that it could open up a program where people can maybe look for laws or principles specific to an operative at the individual cell level and try to mix them or augment them with mechanisms. So, because of our time, I think I'll just stop here. But basically, the main program that I'm interested in, and I think this work on protein-protein interactions is a part, is to really look for a research program where mechanisms and laws, and I should also add this quick note, by laws, we shouldn't think about strict laws of, fundamental physics. Laws as in generalizations or patterns or principles. Any of these kinds of generalizations that help with answering some kind of a why question. This program of looking for mechanisms and laws and how they can augment explanations in cell biology, I think has a lot of potential. And in these two papers that I've cited to the right, I've tried to look at some specific cases where a program focused on mechanisms and laws can be fruitful in cell biology. Thank you very much, and I'm going to stop the recording now.